Benissimo, allora grazie per essere qui, grazie per la partecipazione al premio Campese dell'ISNAF e innanzitutto congratulazioni ai finalisti perché siete stati scelti tra dei concorrenti che erano molto quotati e molto preparati. Quindi già essere finalisti dovrebbe essere considerato un risultato già raggiunto e notevole. Quindi adesso vorremmo noi della giuria sentire la vostra presentazione e poi decideremo su quello che sarà il vincitore per il premio. Ma in ogni caso vi vorrei dire a nome di tutti che sia noi che siamo i tre revisori che partecipano al premio, che tutti i revisori che hanno partecipato alla valutazione delle domande sono rimasti molto colpiti dalla vostra preparazione, da quanto siete dedichi al vostro lavoro e dal futuro molto brillante che avete davanti. Quindi non so se il dottor Dalla Favera o la dottoressa Colla vogliono aggiungere qualche altra considerazione.
No. Grazie Alessandra, niente da aggiungere, anch'io ho notato ottimo livello dei partecipanti.
Io mi allego agli altri due revisori.
Benissimo.
Excuse the interruption. This is Cita Zufada, the president of ISNAF. Can we please switch to English?
Oh, okay, sure. Sure, I wasn't sure about that, but yeah, we can. Actually, my Italian sounded funny, so English is even easier. So yes, so here we are for the presentation of the finalists for the ISNAF Award, the Paola Campese Award for Research in Hematological Malignancies. The three finalists are Dr. Casirati, Dr. Corinaldesi, and Dr. Gurnari. And we will have the presentation to go in the alphabetic order. So the first presenter will be Dr. Casirati. Dr. Gabriele Casirati, please go ahead.
Thank you, Alessandra, for this fantastic introduction. I will now share my presentation. In the meantime, I just want to say that it's really a great pleasure and an honor to be here tonight to present this work to the ISNAF finalists and to the jury. So without further ado, I will proceed and I will talk tonight about my project on epitope editing to enable targeted immunotherapy of acute myeloid leukemia. And then I'll talk about some potential further developments. So as you probably all know, acute myeloid leukemia is one of the most common acute leukemia in adults. And despite several recent agents that have been approved for clinical use, and despite the diffuse use of allogeneic hematopoietic stem cell transplantation, the majority of the patients are faring quite poorly with an overall long-term survival of less than 30%. And while we have seen great advances in the field of targeted immunotherapies for other leukemias, in particular for hematological malignancies, there have been no such thing for AML, which has lagged behind other hematological cancers. And if we look at the FDA-approved therapies for AML in the last 50 years, we don't see pretty much any targeted immunotherapy, except maybe for the introduction of gentusumab ozogamicin, which is just an antibody-drug conjugate, but nothing especially in the context of cellular immunotherapies. And this is particularly due to the fact that we have quite a lack of stringent leukemia-restricted antigens. So there are no antigens on the surface of AML cells that may be safely targeted without incurring on-target of tumor toxicity. This is simply related to the fact that AML cells are very similar to hematopoietic stem and progenitor cells, or they express antigens that are also expressed on mature myeloid cells. And even though you may be able to survive without issues, B-cell aplasia induced by CD19 CAR T cells, for instance, you cannot survive long-term with myeloid aplasia or even general hematopoietic aplasia induced by depletion of hematopoietic progenitors or stem cells. So there are a number of possible approaches to solve this issue, and one is simply to limit in time the exposure to the targeted immunotherapies. And this has been definitely proposed and even applied in clinical trials, but reducing the exposure to the immunotherapy is likely insufficient for disease eradication. So you're going to reduce your chances of a cure. On the other end, there's been a very elegant solution proposed by the group of Sargil at UPenn, which entails the genetic knockout of the target antigens on transplanted HSPCs, therefore making them invisible to immunotherapies. But again, this approach is only possible for antigens that are irrelevant for hematopoietic development. And so we also think that these are antigens that are typically less relevant for leukemia biology overall, and therefore they're more likely to be lost by downregulation. So what we thought was to instead introduce the idea of epitope editing, which is simply the removal of the target epitope without the need for gene knockout. And this is simply the single amino acid changes in the extracellular domain of the target antigens on the surface of hematopoietic stem and progenitor cells that are used for bone marrow transplantation. So we would work on donor cells that are, for AML, typically allogeneic, and make them completely invisible and not recognized by the targeted immunotherapy, but this time without any effect on target expression, regulation, and function. And this makes it possible pretty much to target any surface antigens on the surface of AML, regardless of the shared expression on healthy HSPCs. And again, by choosing functional antigens, we believe that this is going to reduce the overall risk of tumor immune escape. So first of all, we decided to go for one antigens, and we started off with FLT3. It's very well characterized for AML. It's a receptor for FLT3 ligand. It is also expressed on a number of hematopoietic progenitors. But importantly for AML, we think from the data we have, it's expressed on the majority of patients and to quite a decent level, and the majority of AML cells, even within patients. And luckily for us, I believe there are also some FLT3 targeted immunotherapies in current preclinical development. So what we did is design a pipeline to screen for the mutations in the extracellular domain of FLT3 by expressing these libraries in cell lines and then staying with two antibodies at the same time to ensure surface expression of the target, identify mutations that abrogate the binding of the therapeutic antibody clone, and then we sequence and revalidate by recloning individually each variant, and we assess very thoroughly its functionality, because again, FLT3 is a receptor, so we assess FLT3 ligand affinity first of all, and then we move forward all the downstream signaling and all the effects on the cell biology, its stability, and so on. So we make sure that the variant that we have selected that is not recognized is fully overlapping with wild-type FLT3. As an example here, we have extracellular domain 4, where we have identified this mutation in position 3 and 9 that is sufficient to abrogate the binding of this therapeutic clone, which is termed 4G8. So we moved forward and again confirmed that this mutation nearly completely abrogates the binding of the therapeutic antibody while not having any effect on FLT3 ligand affinity or the downstream signaling by looking at kinase phosphorylation. But we run several additional assays to confirm that we don't see an effect on the proliferation, transcriptional signature, and so on. But most important, we also transplanted these cells in vivo, and we confirmed that they are similar to non-native cells. But obviously, when we talk about stem cells and an actual therapeutic strategy for patients, we want a technology that can introduce this mutation safely into hematopoietic stem and progenitor cells. And luckily for us, by looking at the nature of this mutation, we were able to do so by exploiting adenine-based editing. And base editing is a recently introduced technology by the lab of David Liu and Nicole Godelli that can introduce single base changes very precisely without the need for DNA double-strand breaks or without the need for a template donor. And we identified a combination of editors and guide RNAs that were able to introduce the intended mutation at very high levels. And again, this was true also in primary HSPCs that could be edited at over 80 to 90%, even in stem cell enriched subsets, so CD90 plus, 45RA minus subset, without any effect on the stem cell phenotype and functionality and differentiation. And again, as I mentioned, if we look in vivo, both in primary and secondary recipients, we did not see any effect on the engraftment capacity, the multi-lineage differentiation, and also the stability of the gene marker of this mutation in vivo. And again, this is very relevant because in mice, FLT3 ligand is fully compatible with human FLT3, so it does reduce signal. So obviously we wanted also to confirm that these cells that we edited were fully resistant to targeted immunotherapies at FLT3, and we did this by preparing co-culture killing assays, both with cell lines expressing the FLT3 variant or even with CD34 HSPCs that were edited or not for the target. And again, you can see here that the wild-type cells are completely eradicated after 48 hours of co-culture, while the edited cells can persist and they still express FLT3 at very high levels. So again, confirming the basis for our strategy. But obviously we wanted to take this a step further, and we moved in vivo with a much more complex model where we wanted to model the occurrence of leukemia in the context of an healthy hematopoiesis as well. So modeling an early relapse after allotransplant, if you like. And so we transplanted MBSGW mice with CD34 HSPCs that had been edited for a FLT3 or a control locus, such as AVS1. And then in the same mice, we also transplanted an AML patient-derived xenograft that, again, could engraft, and together with the cells, we had both at the same time, and then we treated these mice with FLT3 CAR T cells. And interestingly, obviously, we observed complete eradication of this disease in the presence of the CAR T, as expected, because this disease was FLT3 positive. But most importantly for our strategy, we also were able to confirm the resistance of the hematopoietic progeny derived from edited HSPCs. And you can see here in the ABS1 condition, the granular monoprogenitors or the lymphoid prime multipotent progenitors that are FLT3 positive were completely eradicated in the presence of the CARs, while if we look here in the FLT3 edited condition, these lineages are still present, and they are present at a similar proportion as non-treated mice. So again, this confirms the viability of this idea, but we again want to go even further, and we started to study different patient-derived xenografts, because again, we know AML is very heterogeneous, and also there's a lot of intra-tumor heterogeneity, so there is always the chance for target downregulation or loss. And we studied some diseases that were partially resistant to FLT3 targeting alone, even though they had some FLT3 expression. So we reasoned, yeah, obviously we will always encounter some leukemia that will resist a simple monotherapy, even if it's a CAR T. So we studied, again, additional antigens with the idea to design a multi-target immunotherapy. And we started off again with CD123, which is the receptor, the alpha chain of the IL-3 receptor. Again, another very well-known target for AML, widely expressed in most cases. And again, here we repeated our work and identified guide RNAs that could abrogate the binding of a therapeutic antibody, which is called talacotuzumab. And again, we exploited this to design CD123 CAR T cells with the same specificity. And in this double reporter model, we were able to introduce the mutation for both epitopes at the same time. And again, by co-culturing all these fractions, so either the unedited one, the single edited for either target, or the double edited, you can see here that the pink line here is the double edited cells that are fully resistant to the dual CAR T cells in vitro. And again, if we took this in vivo, and this time we treated the mice that were co-engrafted also with that PDX that was resistant to FLT3 monotherapy, FLT3 CAR T cell monotherapy alone. And in this case, by using both CAR T cells at the same time, we were able to eradicate this disease. And again, looking at the healthy hematopoiesis all across all the different lineages, here it's shown in absolute count, because again, by using different targets, you sort of fake the proportions, because C123 depletes more myeloid cells, while FLT3 depletes more lymphoid progenitors. And again, you can see here that double edited HSPCs and their progenies show resistance to the effect of the dual CAR T cell therapy. So in conclusion of this part, I just want to show that epitope editing confers this resistance to CAR T cells, generating an artificial leukemia-restricted antigen. And this can be achieved by adenine-based editing with minimal genotoxicity on hematopoietic stem cells. And these stem cells have preserved the stem cell repopulation and differentiation capacity also in vivo. And most importantly, for the success of AML immunotherapy, you can do this with multiple antigens, and so you can devise multi-target immunotherapies. Just a quick look at what we can do also with this approach. I just want to take a few words more about the bone marrow transplantation, and in particular conditioning. We know that we use chemo and radiotherapy to condition the host bone marrow but obviously this is associated with a number of toxicity. So in recent years, we have seen a number of groups working on targeted immunotherapies, and in particular monoclonal antibodies against the HSPCs to deplete them. But we know that this strategy, so non-genotoxic immune-based conditioning, is associated with a number of limitations, in particular because of the antibody pharmacokinetics, which may result in incomplete myeloablation or even graft failure. So in this story, we exploited epitope editing instead to protect the graft cells to make them invisible again to the conditioning antigen, making them resistant. And in this way, we can achieve in vivo selection of epitope-edited cells and devise novel immune-based conditioning regimens and transplantation strategies. We worked on c-Kit, which is another very long antigen for stem cells. We repeated all our work, identifying mutation and validating this. And we also performed a competitive transplant in vivo where cells that were either edited for the c-Kit epitope or again control cells were also marked with a lentiviral vector expressing different fluorescent proteins, so we can tell them apart. And then we administer different antibodies regimens with different distribution, but with the same cumulative dose to see if we can achieve better in vivo selection. Just to show a couple of results, I want to be brief because we have a little time. This is the unedited, the untreated mice where we have the blue cells which are edited and the green ones instead, which are the unedited ones. So they're not protected. And when we start to apply our immunotherapy, you can see that progressively we achieve more and more selection in vivo across all the different lineages, and in particular more for the myeloid ones. But if we even distribute the dose more, so same cumulative dose, but across a longer time, so we expose the cells for a longer time to lower antibody doses, overall, we achieve a near complete selection for the edited cells, which again are displayed in blue. So they are fully protected, and we can get over to 80% in the progenitors and the myeloid cells. Why is this? Again, simply because the selection is proportional to the KIT expression levels as expected, so only the stem and progenitor cells are KIT positive, and the myeloid and the erythroid cells, while the lymphoid cells do not get selected, but they get only replenished from the HSPCs if you give enough time. So in conclusion, I just wanted to say that also key additional targets can be epitope edited, and this can be used to protect HSPCs from monoclonal antibodies that may be used for in vivo conditioning, and particularly non-genotoxic conditioning. And this enables us to select for these cells in vivo, and this opens a number of different possibilities for allogeneic or autologous hematopoietic stem cell transplantation, both in the benign and malignant setting. And finally, that these low-dose distributed antibody regimens are more effective at achieving in vivo selection. So finally, I just want to acknowledge all the people that have worked in these projects, and particularly Pietro Genovese, my mentor, all the people in the lab, and all of you for your attention. Thanks.
Thank you for this extremely interesting presentation. Now we can open the floor for additional question and discussion. And while everyone is thinking, I have a question for you. So you showed two possible avenues for this discovery, the one of direct targeting of the FLT3 positive AML, and the one to use it as a novel approach for conditioning regimen. Which one do you see is going to be moving first in the clinical scenario, and also whether you think that there are already some identified good partner for your approach? Because as we all know, we almost never do monotherapies. So for example, do you see that your approach for the FLT3 positive AML will be potentially combinable with the bispecific monoclonal antibody, or do you think it will be more combinable with a classic either targeted therapy or chemotherapy?
That's an excellent question. First of all, which is going to move to the clinics first? It's difficult to tell. What we were thinking to do, and we're trying to pursue now, is to first translate the AML immunotherapy, simply because it would start with safety in an allogeneic stem cell transplantation, so that it's easier to manipulate the donor cells, and because we would treat patients that obviously we will start with second line relapsed patients. So obviously we will start with an innovative therapy, but obviously in patients that have a potentially poorer prognosis. And we are discussing this with the company that owns the IP for the 4G8 antibody to move this forward. We are also in contact, and we will probably pursue this academically in collaboration with the group of FoodCheck in Germany, because it's currently preparing a clinical trial with the 4G8 CAR T cells. It will be the first in human clinical trial for these CAR T cells. So this will be the first proof of concept, and then the second step will probably go for the epitope editing. On the other end, about the conditioning and protection, there is an extreme interest, especially from industry, because especially now we've seen a huge interest in the sense of in vivo gene therapies and in vivo delivery. And what people are particularly interested about is combine this mutation with additional therapeutic mutation and achieve in vivo co-selection of the edited cells. So you can reach therapeutic thresholds, and you can have less toxic transplants, because you don't need to administer the transplant in a short time period, and you remove a lot of toxicity, even reduce aplasia. So there's a lot of interest in that sense, especially in the gene therapy space, because people would like to use this combined with potentially therapeutic gene therapies and modifications.
Great. Any question, Riccardo, Simona, or Cinzia?
In terms of gene therapy, how safe is gene therapy here? So when you edit your cell, did you check if anything else happened? So this is very, of course, is the concern of all the world, particularly, for example, sickle cell anemia. This happened, and I'm particularly interested in sickle cell anemia, so I know that this is what is happening right now. So do you think that you are able to really editing this cell in a way that you are not able to induce a secondary alteration, deletion, gene? Is that just a question?
No,
I think
I understand. No, it's very
innovative, and I congratulate a lot. It was just a question.
No, it's a very hot topic recent times, especially because we're starting to see what are the potential consequences of gene therapies and gene manipulation on stem cells. We've seen a number of issues. Just a couple of weeks ago, there was the publication of the New England Journal of Medicine about a gene therapy that resulted for lysosomal storage disease, for adrenoleukodystrophy, which resulted in a patient that evolved to AML in nearly 10% of the treated patients. But again, there was a lentiviral vector with a strong viral promoter. So that was a totally different story. And we've seen some issues also from the trials run by Matthew Porteous with CRISPR and sickle cell disease that resulted in failure of engraftment or delayed engraftment. So we know that there are significant risks. But what I can say here is that the technology that we are using for these approaches is base editing. And base editing is among the safest of all the genome engineering technology. And this is due to a number of reasons. First of all, it's also adenine base editing, which is, again, among base editing, the least genotoxic. And this is due because the CRISPR system just targets the deaminase to the region that needs to be modified. And there is no nuclease involved in some, so there is no double strand break. You don't induce any risk of large deletions. You don't risk even small deletions, and there's actually next to zero risk of translocation. The only effect is due to the deaminase, which is very localized, and we are even started. I have another project that I'm running that is specifically devised on evolving additional deaminases and with a more safe profile that is even more restricted to reduce even on-target bystander mutation. So among all the technology that we tested, this is the safest of all, and that has a very minimal effect on stem cells when performed ex vivo. And we know that we have partners that are testing this also as an in vivo delivery system using LNPs and viral-like particles. And in our experience is the one that, as far as we know, affected the least the safety of the cells. And the other part is the validation of the mutation, but that is what we really put a lot of effort in. And in the publication, we really tried to build a strong story that these mutations are fully safe to be edited in stem cells. The FLT3 is even present in non-human primates, so it's pretty much evolutionarily conserved. And the KIT is a mutation that is found in mice, for instance. So in regions that that are not close to the ligand binding site. So we have really no concern about the mutation, and they're not present in databases associated with cancer. So in any case, we have a number of both in vitro and in vivo validation that confirm that this mutation is safe, and the technology is one of the best that we know of. So for now, I think we are pretty sure we can work with this, and it's probably better than what we've seen in clinical trials for now.
Great.
Thank you. Thank you very much.
How are we doing with time, Dr. Zuffa?
Exactly. Oh, time, I was addressing time in my question in a clinical setting. With the, can I have time for a question?
Just 1 minute.
Okay,
Quick question.
quick question.
So the ideas are brilliant. In practical term, they are all custom made from patient cells, both the HSC and the T cells. So I wonder, how does that play with the timing needed to prepare the reagent, and whether the brilliant idea of epitope manipulating HSC can better be combined with bispecific, trispecific.
In terms of combining using with bispecific antibodies or other formats, it works perfectly well. There's no difference in the protection that you would expect. And we've seen this. It's been a number of other groups have published after us on the same approach. There's one of Lucas Yeager from Basel that has recently published a Nature on CD45 epitope editing and used an ADC, for instance. There's the group of Sargi, UPenn, that again published another CD45 paper with epitope editing, and they use also BiTEs. So the concept is it's the same. First of all, we use CAR T cells because we thought they were the most effective and more stringent in terms of efficacy. Timing. So in short, you're right. Obviously, there's the time needed for the manipulation to the manufacturing. The only product that, strictly speaking, needs to be from patient matched or actually derived from the donor is the HSPC manipulation. The first clinical trial, especially if it's academic, will also use T cells from the same patient with the same affinity that simply will be split, and the stem cells and both T cells and stem cells will be manipulated and frozen before administration, simply to allow for quality control and product release. Again, this is sort of compatible with the normal timing for allogeneic stem cell transplantation, which again usually is not short in any case, especially unless the donor is a relative and is already been identified. It usually takes, in the case, 2 to 3 months in average to get to the patient to transplant. So adding the 10 days for T cell and HSPC manipulation should not be an issue. On the other end, the longest protocol for now is T cell manipulation, and that, strictly speaking, does not need to be from the patient, because we have now seen a number of clinical trials, especially the ones run by Wasim Qasim from UCL, that produces CAR T cells from healthy donors. So they are off the shelf, and they are modified to avoid GVHD. So they have either T cell beta knockout and so on. So we believe that the field will move progressively forward towards either in vivo the generation of CAR T cells, probably not for AML, not for transplantation settings, but also much more closer to off-the-shelf therapies. And I think that is probably going to be the way for AML.
Thank you very much, Gabriele. Thank you, Dr. Casirati. We will now move to the next candidate, that is Dr. Corinaldesi. So Clarissa will be presenting her research for us.
Can you see my slide?
Yes.
Perfect. So, thank you, first of all, Professor Ferrajoli, for the introduction, and I want to thank the scientific committee and the organizers for this opportunity to present some of the results of my research today. It is an honor for me to have been selected as a finalist for the Paola Campese Award. So today I'm going to present intra-tumor heterogeneity and markers of resistance in pediatric Burkitt lymphoma. Burkitt lymphoma originates from the malignant transformation of B cells in the germinal center and is the most rapidly proliferating human cancer. The World Health Organization recognized 3 clinical variants of Burkitt lymphoma, the sporadic, the endemic, mostly associated with malaria and Epstein-Barr virus, and the immunodeficiency associated, that is mostly related with the HIV infection. Burkitt lymphoma is the most frequent B-cell lymphoma in pediatric patients, accounting for more than 80% of the cases. And although more than 95% of them achieve remission, the prognosis for non-responder patients remains very poor. The genetic hallmark of Burkitt lymphoma is the chromosomal translocation of the MYC oncogene under the control of regulatory regions in the immunoglobulin loci, leading to ectopic MYC expressions. Other genetic lesions have been recognized. All of them have the goal to hijack the germinal center B-cell physiology. So as I say, Burkitt lymphoma derives from germinal center B-cells, so I'm going to introduce quickly the germinal center. Germinal centers are histological structures that form in the secondary lymphoid organs after that mature naive B-cells encounter an antigen for the first time, and the goal of the germinal center is to produce memory B-cell or plasma cell that are able to produce antibody with high affinity for the antigen. In the dark zone of the germinal center, B-cells proliferate and undergo somatic hypermutation of their immunoglobulin variable region in order to increase the affinity for the antigen. And in the light zone of the germinal center, these cells will be selected for this affinity. They can re-enter in the dark zone of the germinal center for further editing, or otherwise they can be selected to differentiate in memory B cells that will retain the memory for the antigen, or they will differentiate in plasma cells that are cells that are able to release this antibody against the antigen. If the affinity for the antigen is lost, the cells will go in apoptosis. So obviously all these genetic editing that are happening in the germinal center are the best case scenario for malignant transformation. The major B-cell lymphoma derive from malignant transformation of germinal center B cells at several stages. Burkitt lymphoma derive from a dark zone B cell. In the past few years, our effort and effort from others has been focused on better understanding the dynamic of the germinal center, and single-cell transcriptomic analysis of normal germinal center B cells helped us to understand this heterogeneity. Here I'm showing a new map of more than 40,000 germinal center B cells. If you are not familiar with a new map, each dot represents a cell. Each cluster that is differently color-coded represents a group of cells that share the same transcriptomic signatures, and closer these clusters are, more similar from a transcriptomic point of view they are. We were able to recognize different population within the dark zone and the light zone, and in addition we were able to identify intermediate population that were showing a transcriptomic feature that were transitioning between the dark zone and the light zone, and we were also able to identify the precursor of memory B cells and plasma cells. If we try to connect computationally this population in order to understand how they relate to each other, we can see that with the pseudo-time analysis, we were able to identify cells that were re-entering the dark zone, or cells that were committed to differentiating plasma cells and deriving from a specific light zone B cells, or cells that were committing to differentiating memory B cells. This data demonstrates that actually the germinal center B cells move across a continuum of states instead than moving between two different compartments. So obviously, single-cell transcriptomic analysis was able to give us a lot of information about the heterogeneity of normal germinal center B cells. So we used single-cell transcriptomic analysis on tumor cells. And the tumor that we select was Burkitt lymphoma. We collect pediatric sporadic Burkitt lymphoma. All of them were EBV negative. We were able to collect frozen Burkitt lymphoma single-cell suspension at diagnosis from 11 patients. And because this is a retrospective study, we know already that 3 of them were not responding to therapy. At this point, we isolate viable cells we check for B-cell representation by fax before proceeding to single-cell RNA-seq analysis. And the goal was not only to have transcriptomic information about these cells, but also this was paired with the immunoglobulin receptor sequence information. As you know, each B cell has a specific and unique immunoglobulin receptor sequence, and this could help us as use this identifier to identify tumor cells. In fact, what we were able to do was to divide the analysis of tumor cells from non-tumor cells, because the tumor cells were sharing the same immunoglobulin receptor sequence. As you can see here, what is coming out from the analysis is that patients clusters independently from one another, suggesting that the patients were distinguishable based on transcriptional features of their tumor cells. Conversely, the same analysis done on non-tumor cells show that the cells were clustering based on their cell type and not based on the cell of the patient of origin. And we were able to identify monocytes and macrophages, B cells, T cells, and also a small population of cancer-associated fibroblasts. So to better understand the heterogeneity of Burkitt lymphoma cell, we decided to merge all the Burkitt lymphoma tumor cells, and after batch correction, we were able to obtain 12 different clusters, suggesting that these tumor cells were really heterogeneous. And analyzing the top 100 upregulated genes in each one of the cluster, we could do a pathway enrichment analysis. And these analyses show that patients were sharing pathways related to cell cycle, to the B-cell receptor signaling, and to the NF-kappa-B signaling. So at this point, what we wanted to understand was what was the relationship of the Burkitt lymphoma intratumor heterogeneity with the normal germinal center B-cell signatures that we already identified in the lab. As you can see in this view map here, more than 50% of the Burkitt lymphoma cells were actually, as expected, associated with a dark zone-like. What was not expected was that the remaining 50% of tumor cells were actually associated with a light zone-like or a pre-memory-like transcriptomic signatures. And as you can see also here in the heat map, you can appreciate some of the genes that are associated with each of these clusters that were upregulated differently in each one of these populations. So the Burkitt lymphoma intratumor heterogeneity resembles normal germinal center B-cell states. The other open question that we have was like, can we identify differences between non-responder and responder to therapy at diagnosis? So the first thing that we want to see was to understand the distribution of these tumor cells from these two different groups of patients across the normal germinal center B-cell signatures. And even though most of the signatures show an even distribution between these two groups, there were some of the signatures that show differences, and in particular, the light zone A population was depleted in tumor cells of non-responder patients, suggesting that even with a small sample, with a small number of samples that we have, we could clearly see differences from a population point of view between these patients at diagnosis. So moving from a population point of view to a gene expression point of view, we analyzed differential expression gene expression between the non-responder patients and the responder patients. As you can see here, the top upregulated genes in the non-responder patients were mostly associated with the B-cell receptor signaling, with the Notch signaling, the TGF or interferon signaling, while the most upregulated genes in the responder patients were mostly associated with cell cycle, light zone, or pre-memory signatures. Our goal was really to understand if we were able to identify markers that help us to distinguish non-responders and responder patients at diagnosis. So we focus our attention on the top upregulated gene in the non-responder group, that is TPM2.
5 minutes left.
TPM2 encodes for the beta-tropomyosin actin filament binding protein family, and the muscle cell has a central role in culture.
I don't see the slides. Do you all see the slides?
Yes, I can see the slide.
Okay, so
While in non-muscle cells, it helps to stabilize the cytoskeleton. When we check the RNA expression of TPM2, we found that, obviously, as expected, it was mostly expressed in muscle cells, dendritic cells, and fibroblasts, but it was not expressed in mature B cells. And when we check for protein expression, again, TPM2 was clearly expressed in muscle fibers and blood vessels, but in the germinal center, the staining that we were seeing, the expression that we were seeing, was mostly associated with dendritic cells or blood vessels. However, when we check in some Burkitt lymphoma cell line, we could clearly see, and consistent with the single cell data, ectopic expression of TPM2 that was detected in a subset of Burkitt lymphoma cell line, both at transcript and the protein level. Obviously, the first thing that we want to do was to check TPM2 expression in our diagnostic Burkitt lymphoma specimen, and we can clearly see that TPM2 was highly expressed in the tumor cell of a subset of patients, and this subset of patients was enriched in non-responder patients. Also, when we check using bulk RNA samples at the TPM2 transcript level, we could see a significant enrichment of TPM2 in the non-responder patients. At this point, finally, we wanted to understand if the expression of TPM2 was associated with outcomes. So both transcript and protein level could discriminate between patients that were associated with the worst outcome. In fact, as you can see here, TPM2 high or TPM2 positive are associated with patients with the worst outcome. In conclusion, what I show you today is that single-cell transcriptomic analysis of pediatric Burkitt lymphoma reveals intratumor heterogeneity, and this intratumor heterogeneity is associated with shared pathways related to cell cycle, B-cell receptor signaling, and NF-kappa B signaling pathways that are mimicking different states of normal germinal center B cells. Therapy responsive and non-responsive Burkitt lymphoma tumors are clinically and phenotypically indistinguishable, but display difference in single-cell RNA identified subpopulations. And also, moving to a gene expression analysis, we were able to identify differences, and these differences were showing TPM2 as a potential biomarker for the early detection of patients who will not respond to therapy. This project was a collaborative project between our group at Columbia under the leadership of Katia Basso and the group of Lara Mussolin from the University of Padova, but was also possible thanks to the collaboration with several institutions in Canada, United States, and Italy that provide us samples and clinical information. And thank you for your attention.
So thank you very much, Dr. Corinaldesi. Now we can open the floor for the discussion. If there are questions for the candidate.
Can I start?
You can start.
I have a few questions. I mean, congratulations, it's a very nice work. One question I did not get, this is by fault, I couldn't follow. When you did the single cell, and the other is curiosity, when you did the single cell RNA sequencing in Burkitt lymphoma, which source of cell did you analyze? Because in normal, you analyze germinal, I mean, at the beginning you show germinal center analysis, but then I doubt that you did germinal center.
Oh.
It was
nodal tumor or a fusion.
Sorry?
Tumor.
Was tumor. Oh, so you look at the tumor and you look at the region of in the tumor. Okay.
Was pleural effusion or nodal tumor?
Okay. Okay, thank you.
Lymph
And
nodes or malignant pleural effusion, correct?
Mm-hmm.
Either lymph nodes or malignant pleural effusion.
Okay, and then you were able there to figure it out where there was the origin based on the healthy germinal center. Understood.
Mm-hmm. Yeah.
And the second question, so you identify TPM2,
mm-hmm.
and you think is a biomarker of resistance to therapy,
mm-hmm.
slightly expressed in patients who, but you didn't show the function. And if you inhibit this gene, have you any functional data supporting the fact that is just a biomarker, or this gene has also a function in maintaining resistance? So as a mechanism, induce a mechanism of resistance, if you inhibit the gene, can you rescue the phenotype? Can you potentially develop a drug, I don't know, to target, I don't know if it is possible to develop a drug for this protein to potentially overcome resistance?
The function of TPM2 is basically associated with the cytoskeleton. It's a cytoskeleton protein. So I think what it helps what it does is basically if TPM2 is highly expressed, there is some kind of problem with the cytoskeleton, and the problem with cytoskeleton can also lead to activation of pathways that are not normally activated. I don't know the inhibition of TPM2. I don't have any data, or at least I don't know any data. What is interesting actually in general regarding TPM2 is that in other tumors like breast cancer, prostate cancer is actually downregulated, while in Burkitt lymphoma seems to be highly expressed, and nothing was known before about TPM2 and Burkitt lymphoma because it's highly expressed in muscle cells and blood vessels. So when we analyze bulk RNA-seq, that signal was diluted, so we couldn't understand that before. But regarding I don't know, regarding Burkitt lymphoma inhibition, I don't know what can happen.
But are you still investigating the function of the gene, or you think that is just a biomarker?
Well, I would like to continue to investigate the function of the gene. It's not my main focus in this moment, but that would be interesting to do.
Thank you. Thank you.
And I have one more question. It also has to do with the kind of pathophysiology. So you notice that the non-responders were depleted in the light zone pro-memory cells. So what is the physiological function of those light zone pro-memory cells that you think could provide an advantage to the responders or a disadvantage to the non-responders?
So the signature that was mostly depleted was the light zone A population, not the pre-memory. So in the light zone A, probably there is high activation of PCR signaling, or so I don't know the answer of your question, but I think that there is an hijack of the normal physiology of some pathways, and this is the reason why they downregulate, or they are depleted of the light zone A. It's like, or they resist more, like they are pro-survival more, let's say. So that could be, I don't know. But I don't know exactly why they depleted light zone A.
Okay.
And also, I need to say that we see that, but that is coming from a very small group of patients, and for sure it needs further validation. And
but you're dealing with a rare disease, so
yes, yeah. Yes, it was very hard to collect.
It's a rare disease that is very generous in terms of tumor mass. So hopefully we will have, a good balance between the two. You will get very good specimens to work on, even if there are going to be a few cases. Yeah.
Few minutes left.
Okay. I think we are done with Dr. Corinaldesi, because I know that Dr. Dalla Favera is familiar with your project. So unless Dr. Zufada or anyone else has questions, I think we can move to the next candidate on time. So thank you, Clarissa. Congratulations and good luck with the publications.
Thank you.
So the next candidate is Dr. Gurnari. And Dr. Gurnari will be presenting his field of research. So Carmelo, if you want to take over.
Many thanks for the kind introduction, and I'm really honored to be one of the finalists of the Paola Campese Award, because I know it's dedicated to the memory of Paola Campese, and you will know why this is very important to me in a second. My project deals with the trajectories of leukemia evolution, and as we can see, an ounce of prevention is worth a pound of cure was recently in Philadelphia, and this is a quote from Benjamin Franklin. So why hematology? It was June 2009, and I was a 19-year-old student at the University of Udine and studying Latin and Greek. Why did I choose to change? And the reason was that I was diagnosed with Hodgkin lymphoma, 4-stage B, with extensive metastasis in the bone, spleen, and liver. And that was the time where I met the first inspirational figure in my career, that was Professor Locatelli. 3 months later, starting chemo, in between the day of the PT scan and the CT scan, I tried to enroll in the University of Pavia and medical program. I succeeded, and I say to myself, after my remission, chemo, radiotherapy, and autologous stem cell transplant, that I wanted to become a hematologist. So in July 2015, you see the picture with him and my parents at my graduation, and then I started a fellowship at Bambino Gesù Hospital with Dr. Bertagna and him. After that, I went to the specialization program at Tor Vergata University under the guidance of my second inspiration, Professor Maria Teresa Gavoso. And then later on in September 2019, right before the pandemic, I decided to have the US adventure and joining the Cleveland Clinic in Ohio, where I did my PhD degree, and then I was later on appointed as junior faculty. So the leitmotif really stems from my personal experience, because what struck me the most was the mother of a 3-year-old child who had a myelodysplastic syndrome. I tried during my presentation to explain this term because there's a lot of my family connected, so I don't want to bore them to death, and I try to use some simplification of medical jargon. I hope the jury is okay with that. So this pre-leukemic condition happened after this first disease that is called aplastic anemia. And the mother, after 2 years of honeymoon of this kid, asked me, "Doctor, could have we predicted this?" And the fact that I was unable to respond to her drove my attention on the mechanism of leukemogenesis. So what drives leukemia? And there are, I think, some particular instances that I focused on during my initial career. A dictionary for this presentation, I want to say that mutation is everything that is divergent from the original genome sequencing, and this is what we try to track or find when we diagnose a patient with a leukemia. HLA is what identifies our cells as self, so not to be attacked by the white blood cells, or non-self, so pathogens, tumors from which we need to be defended. And bone marrow failure syndrome are a group of blood disorders that are characterized by low blood cell counts, so anemia, low platelet counts, and a tendency to progress to leukemias. And this is what is crucial for the importance of this field, the leukemic evolution topic that I chose for my PhD. Because bone marrow failure syndrome patients have an increased rate of a late clonal complication. They tend to have an increased risk to leukemia that is in the range of 25% at 10 years. These leukemias have a serious prognosis, poor survival. We don't have predictive risk factors, and there's no tailored therapeutic approach. So the study of these patients can really help understanding the mechanism that are generally driving leukemias. So what I did in 2019 was leaving Rome, going to Cleveland Clinic, Ohio, and since aplastic anemia, bone marrow failure syndrome are rare disorder, the incidence is in the range of 2 per 1 million. I then tried to be be collecting data from other pioneering institutions in Brazil and in France to collect the largest series so far. But of course, like the journey wasn't easy. I come from Calabria. I didn't even know what snow was, and instead in Ohio, I understood what that is. And then pandemic and other social difficulties that I had to face in order to then try to succeed and gather more than 1,000 patients, so the largest cohort so far described, which we characterized. So the first thing that we did is trying to understand if this patient had clinical features that were identifiable as predictor harbingers of leukemic evolution. And indeed, we noticed that patients who did not respond to the first-line treatment or that were older than 35-year-old, the diagnosis were the ones who tended to progress more. And this was then also confirmed by our statistical analysis. Important is that when we looked overall at the presence of these mutations at the bone marrow failure onset, overall, this was not statistically significant. Now I want to show you the characteristics of these patients that evolved. So 94 patients out of these 1,000 patients evolved to leukemia, slightly more male than females, with a median time of 4.5 years. Overall survival was 40% at 5 years and was strictly dependent on the number of leukemic cells in the bone marrow, which we call blasts, as in the medical jargon. And again, we noticed that these patients were indeed those that responded poorly to the first-line treatment and were those who had the most severe form of bone marrow failure syndrome. Then we try to look at the molecular features. So we ask ourselves, are all these leukemia arising post-bone marrow failure syndrome the same? Indeed, we were able to find some molecular footprints, and some were also informative of prognosis of this patient. In particular, we have seen that there was a group that was strongly characterized by specific cytogenetics or chromosomal alteration or specific type of mutation that were linked to also some specific genomic signatures. Then we try to understand if leukemia arising post-aplastic anemia was similar to that doesn't arise from aplastic anemia. So we use a statistical sophistication called match pair comparison, which basically tend to adjust for some variables, some factors which we call confounders. In other words, we were able to, this is the statistical method that make us compare apples with apples. And what we found from this is that, again, the leukemias arising post-aplastic anemia were characterized by specific mutation, ASXL1, RAX1, and also specific cytogenetic aberration, like, for instance, the alteration of chromosome 7, and were also in higher risk according to the type of the risk stratification tool which we use in this type of leukemia. After that, we did a cross-sectional analysis, which means we took samples drawn at bone marrow failure diagnosis, during follow-up, and during leukemia evolution. And we studied a variety of molecular alterations which we thought were important, crucial for understanding the mechanism of leukemia. And we saw that 18% of patients were already harboring this leukemia alteration at leukemia onset. And you see in the lower left panel that this myeloid these mutations were also increasing, not only in terms of number, but also in terms of size, what we call the variant allelic frequency. So they increased over time, overwhelming the bone marrow. And then we were able to understand which patients, according to the molecular alteration at diagnosis, tend to progress more. In particular, we found that those that already had this myeloid driver at the onset were those who didn't increase risk. And this is a take-home message from this study that you can see from the cartoon, that when we treat a patient with aplastic anemia with first-line treatment, what we create is something that might be then important to study, and in order to understand if the patient has an increased risk from a molecular point of view to go to leukemia.
5 minutes left.
What are my future plans? So I got a grant from the Evans MBS Foundation, which will help me understand why the immune system's checkpoints fail in in stopping the progression of pre-leukemic to overt leukemic condition. And I focus my attention also on other form of bone marrow failure syndrome, in particularly with this novel disease that I think is emblematic of the link in between inflammation, leukemia evolution, and bone marrow failure syndrome. And this is called VEX. So this is an acronym, as you can see on the left. It really struck my attention because is a good example of how we can understand a little bit better and try to prevent leukemia evolution of this patient, perhaps not only with a better molecular characterization as we did before they go to leukemia, but also maybe with transplant approach, as we have recently demonstrated with the European Bone Marrow Transplant Society, with a 2 years overall survival of 75%, demonstrating also molecular clearance after transplant of the pathogenic mutation and reversion of the clinical phenotype of this patient. And last but not least, the other important aspect that I'm focusing on is the familial leukemias, because inherited predisposition to leukemia is not only an issue of pediatric patients, as we thought until maybe 10 years ago, but now we know that 1 out of 10 patients with the leukemia have a familiar predisposition. So my focus is now try to understand what are the early identification strategies, surveillance for relatives, and also address an important gap, which is how to select donors. And this was the object of another EBMT effort. Last, I want to really acknowledge my colleagues, my family, who is always supportive wherever I go in the world, and my mentors, Professor Franco Locatelli, Maria Teresa Vosa, Jaroslav Maciejewski, because as William Arthur Ward said, the mediocre teacher tells, the good teacher explains, the superior teacher demonstrates, but only the great teacher inspires. And I really am grateful, and I really think that they inspire me the most. Thank you.
Thank you for this very nice presentation, this journey through your life and your career that is really quite impressive, Carmelo. So the floor is now open for discussion. And while Dr. Colla warms up, because I know this is a field where she's going to have some questions, not that Dr. Della Favera won't, I'm asking you a question. In terms of the aplastic anemia patient that then developed the MDS, do you think there is an option to intervene before we give them aplastic anemia treatment, or do you think the train is already set and we can only act later and maybe direct those patients toward the stem cell transplant? This is, I know, a very clinical question, but.
Yeah, and thank you. And actually, this is precisely the focus of the research and like the gap that we have. So when we give the immunosuppressive treatment, aplastic anemia is like an autoimmune disease, as you know. So we need to tame the lymphocyte attack to the bone marrow. We create iatrogenically what Seishogawa called a bottleneck effect. So we depauperate the already depauperated the bone marrow, and we only few healthy hematopoietic stem cells survive after the immunosuppressive treatment. If in those there are some mutations, so this means that like the repopulation of the bone marrow becomes oligoclonal and then perhaps clonal. And indeed, we know from studies like mine and others that clonal hematopoiesis is the rule in bone marrow failure syndrome. Almost 70% of patients have either myeloid clones, PNH clones, or HLA lacking clones, which is a particular and important feature. So I think that if we sequence this patient for this triad and we have this triad of information at the beginning of their journey, we can definitely tailor the treatment and perhaps think for early transplant strategies if possible.
Well, we often have the dilemma at presentation whether to have a direct stem cell transplant or try ATG cyclosporine, like you said. So this could be potentially useful. Now, Dr. Colla and Dr. Dalla Favera, I don't want to take all the time.
Well, I have to say that I know very well Carmelo's work is becoming a leader in the field. We were together in Philadelphia, so I'm very familiar to his work. So congratulations, not just for your work, also for your story. I think, I mean, you did an incredible work. You are talking about more than 1,000 patient sample. And this, of course, this database, in my opinion, can offer to investigate many different questions. And one out of my curiosity is this. So you are talking about a familiar mutation. I would talk about predisposition mutation, somatic mutation. Sorry, germline mutation. Could you look for germline mutation in your database and check the correlation for the somatic mutation? For example, I'm talking about this because in the field of telomere biology, this is my work. Unfortunately, I mention my work, but it can be true for many other. So, for example, patient with a germline mutation in telomere maintenance genes specifically acquired specific mutation, for example, in splicing factor U2F1. Can you investigate your predisposition, let's say patient, find the predisposition mutation, the germline mutation, and see the association? This can give you the possibility to possibly have some biomarker to check for patients with specific germline mutation. For example, now for patients with telomere biology disorder, we know if they acquire U2F1 mutation, this patient, they are going very badly, need to be monitored very strict, you need to do something. While if they acquire mutation, for example, in POT1, they don't do anything, it's a genetic rescue. So maybe you could interrogate your huge database and also find a new other question to answer, because this is incredible data set.
Thank you, yes.
Can be very clinically relevant in terms of predisposition, in terms of which are the patients who are predisposed to become leukemic soon because they acquire this specific, in that specific context. Because if some specific somatic mutation acquired in a different specific germline context do not do anything, but in that one specific context, they work together, and then they drive the disorder and the progression very quickly.
Yeah, I think this is like a relevant topic of interest, and we tried to do something actually. So we noticed and we were able to confirm that patients, for instance, with GADA2 tend to like have STAG2 as a second somatic alteration, for instance, or patient with SAMD9 preferentially acquired chromosome 7 abnormality or UPD of the same chromosome in what we called somatic gene rescue. So it's basically the vis sanatrix nature, as Lucretius said. No, it's like the nature that tried to rescue itself. And this happens also in telomere biology disorders, as you were saying. We noticed, for instance, some patient with telomere biology disorders that acquired splicing factor, in particular E2F1, and also TP53, for instance. So definitely might be a good thing to do, and thank you very much for
also this can give you the molecular mechanism through which the germline mutation predispose, and also the mechanism of the somatic mutation, why the somatic mutation is occur, give you the mechanism. For example, U2F1 in the context of telomere biology disorder shut down P53 pathway. And so you have...
Thank you very much. Yeah, it's very
good, very nice.
Thank you very much.
So thank you, everyone. I would like to see if Dr. Zufada has any closing remarks maybe to make to this part of the meeting.
Thank you, Alessandra. And I want to start by thanking the finalists. I really enjoyed this. Coming from a background in physical sciences, of course, I'm not a life scientist, but I am in awe for what you do. You know, the award named after Paola Campese is our longest tenured award, right? The first award of the series Young Investigators. And it was initiated by the former president of ISNAF, Professor Campese, in remembrance of his only daughter who succumbed to leukemia. And so we are all in a sense, you know, as ISNAF, as the society that has such a large pool of life scientists, it's only natural that we should have one award that recognizes the tremendous contributions that the Italian community in North America in life sciences has and achieved. And seeing you, the three finalists, have made such important contributions in your early career is satisfying for me as the president of the organization. Congratulations to you. I want to thank the chairs of the award, Professor Ferrajoli and Dalla Favora, and Professor Colla in the jury for an outstanding work. I'm sure that you are not done, because you have now the task of selecting the winner out of this trio of fantastic people. And that's really your responsibility to carry out. And to the public who has participated to this, I am sure they are equally pleased and interested that we have you know, outstanding contributors in our community. So this comes to a close. Thank you all for participating to the symposium. I look forward to seeing you all in Washington, D.C. on November 14th. Have a good rest of the day.
Thank you, everyone, and have a good evening.